everyone and welcome to the internet. What we're going to be doing in today's lesson is having a look at how we work out the outcome of genetic crosses. So hopefully by the end of today's lesson you're going to be able to actually describe how we predict them and then calculate those different genetic cross outcomes. To start off though, we've got 3 questions based around our previous lesson, so have a go at answering these questions in your books for me. So I'm going to give you about 3 minutes to do this today. Let's check those answers then. So for question 1, we know the total number of chromosomes in a body cell is 18, so to work out the number in a gamete we divide that by 2. So 18 divided by 2 gives us 9. Number 2, the answer is meiosis. And for number 3, a range of marks available here, so give yourself a mark for any of these points that you've got. First one, the DNA unzips, or you could have said the DNA unwinds. Second mark is for saying free bases pair up with the complementary base pairs. A third mark if you actually named those complementary base pairs, so A and T and C and G. Fourth mark is if you said the chromosomes line up along the equator of the cell. If you said the middle of the cell, that would be acceptable. Fifth mark is for saying the spindle fibres pull them to the opposite poles or the opposite ends of the cell. Sixth mark, the cell divides. Seventh mark, if you said it's by cytokinesis. Eighth mark, if you talked about the fact that the chromosomes line up along the equator again. Ninth mark, if you said the spindle fibres pull them to the opposite poles. 10th mark, cells divide. And the 11th mark, to form 4 haploid cells. So give yourself a total out of 13 for your answer there. Now, as we've already said in some of our previous work on genetics, then what you actually have are 2 copies of every single gene. You've got one from the mother and one from the father. Now, the genes that you've got from the mother and the father, they may be the same, or they could be different. And what we actually refer to, we don't call them genes at this point, we refer to these different forms of a gene as an allele. So you can see the spelling of it in our 3rd point there. So wherever we see the word allele, that is a different form of a gene. So we've got a gene for eye colour, for example, and then we've got alleles, so the different forms of that gene for brown eyes, blue eyes, green eyes, etc. So the allele is the different form of a gene, and the gene is literally the section of the DNA that codes for a particular characteristic or protein. So make sure you've got those 3 points written down. The first thing we need to understand about these alleles then is the fact that when we're looking at them, we find that they're either dominant or recessive. If we have a dominant allele, then if we have either 1 or 2 of those alleles present, then that characteristic is the one that's shown. So if you've got 2 dominant alleles or just 1 dominant allele, that characteristic will be shown because it's dominant. It's going to technically overpower the other one present. And the way we show this when we come on to do our genetic diagrams is by using a capital letter for it. The other one, the recessive allele, this is a characteristic that we will only express if both of the alleles are recessive, and we use a lowercase letter to represent that one. So make sure you've got these next 2 key terms written down. One thing you're going to notice in this lesson is we've got a range of new terms we need to know. So this is a great way to start learning them by using your flashcards. So on one side of your flashcard, put the term, and on the other side, put down the definition. And then you can just put them into a pack and then just test yourself over and over again. Because if you don't know the terminology and what it means, you've got no hope of answering the questions correctly. So I'll leave these up for another minute for you to write those down, and then we'll move on to the next set of our definitions. So the last 4 definitions we need to know for today's lesson. Genotype is our first one. Now the genotype just tells us the combination of alleles that are present in an organism. So it will tell us whether we've got one dominant and one recessive, whether they're both recessive, whether they're both dominant. So the genotype literally tells us the alleles we have. Second term, homozygous dominant. So this tells us that there are two copies of the same dominant allele present. So the way we can work this out, dominant obviously tells us it's a dominant allele, and homozygous, break that word down, okay? Homo, same, okay? You can think of it in terms of heterosexual and homosexual. Homosexual people, same sex attraction. Homozygous, same alleles. So what we see here is homozygous dominant, two copies of the same dominant allele are present. And that will be shown by two capital letters. Homozygous recessive just means we've got two copies of the same recessive allele present, and so we would see two lowercase letters. And then finally, heterozygous, it just means our two alleles are different. So you've got one dominant and one recessive allele present. So I'll leave these up for a couple more minutes so you can write down all those definitions. Dead easy little task for you here then. What we've got in the picture underneath are dominant characteristics on the left and recessive characteristics on the right. All I want you to do is pick 4 dominant and 4 recessive characteristics and put them in a list for me. You can pick any 4 from each picture that you want. What we're going to go on to now for the main part of this lesson, now we've got those terms sorted out, is how we actually work out what the offspring would inherit. So the way we do this is through a special kind of diagram, a genetic diagram called a Punnett Square. Now, what I've given you here are kind of like the 6 steps that we're going to go through to make sure we get it right each and every time. So it's kind of like your how-to guide of how to do this. So step 1 is we state the phenotype of both parents, and the phenotype is just the characteristics they show. Second, we state the genotype of both parents, so we'd actually write down those alleles there. Third one, we state the gametes for each parent, so we split the genotype into the 2 gametes. Then we draw the Punnett Square. Step 5 is we then state the phenotype for each offspring, and then step 6 we state the percentage of each genotype present in the offspring. So I'll leave these up for a couple of minutes so you can write down the instructions, and then what we're going to do is actually go through an example so you can see exactly how we do this. So, kind of question we could see is one like this. Work out the genetic cross between a mother who is homozygous recessive for blue eyes and a father who is homozygous dominant with brown eyes. So what we do first of all is we write down the phenotype of our parents. So the mother in this case, the phenotype, what the characteristics she shows, is blue eyes. And for the father, it's brown eyes. So we get that from the question. Then what we need to do is write down the genotype for each parent. So we can see that the mother is homozygous recessive. So that will be the 2 lowercase letters, and we've used B's here because we're talking about brown eyes and blue eyes. And then the father is homozygous dominant, so 2 capital B's. I'd suggest you write this down in your book so you've got a nice worked example to then look back to when you come on to your own questions in a moment. So I'll leave this up for another minute to give you a chance to write it down. The next thing we've got to do is the gametes. So we've got our little Punnett Square set out here, which is pretty much a box. Now what we've got is the father is on the top and the mother is down the side. So we've taken the two alleles and we've put them at the top of the two columns for the father and then put a circle around it. That's how we show it's a gamete. And then for the mother, we've put the two alleles on the left hand side at the start of the two rows and again put a circle around it to show they're the gametes. So again, do a little sketch of that in your books. I'll leave it up for another minute. The next step is we fill out the Punnett Square. Now what I've done here is I've added colours to help you see where they come from. And all we really do is whatever is at the top of the column, we fill into the two cells beneath. So you can see that first capital B in red, we've put it into the two squares just beneath it. We do the same for the capital blue B, two squares beneath that one. And then whatever is at the start of the row as the gamete, we put in the two cells to the right. So the green lowercase b goes into the top two there, and the purple lowercase b goes into the bottom two. That then gives us the 4 potential combinations of the alleles we would see in the offspring. So again, I'll leave it up for a minute to give you a chance to jot those in. The next stage is we need to write the phenotypes for the offspring, so what characteristics they show. Now, if we look at the top left box, we've got a capital and a lowercase. So because we have a capital, the dominant trait is shown, and that was brown eyes. For the top right box, again, we've got a capital letter there, so it would be brown eyes. Bottom left box, we have a capital, so it's brown eyes. And the bottom right, we've got a capital, so it's brown eyes. The only time we would have blue eyes is if both of the B's in the box were lowercase b's. So add in those 4 browns to your diagrams. The final stage, if you remember, is we write the chance of having the characteristic. So in this case, it's 100% chance of brown eyes. Now you could do it as a percentage, as a something in something, or a ratio, or a fraction. It's completely up to you how you write it. But 100% chance of brown eyes, or a 4 in 4 chance, or 4 to 0, or a 4/4 chance. So make sure you've got that last line written in your books. Okay, your turn to have a go. So new question on the screen, have a go at answering that in your books. Just work through the steps as we did on the last example. So let's have a look at this question then. Green pea pods are dominant. So that's our first key bit of information, is that green is dominant, and then we've got yellow as recessive. Work out the possible offspring for a genetic cross of a homozygous dominant mother and a homozygous recessive father. So what we're going to do is draw out our Punnett Square first of all. Now what we can do is actually quite a rough little sketch of these. They don't have to be immaculate when you do them in an exam. My dodgy window is absolutely fine for this. So what we're going to have is we'll put the mother at the top and we'll have the father on the side. Now it tells us up here that the mother is homozygous dominant and green is dominant. So that means the mother must have two dominant alleles. So each individual will be a capital G, remember. So that's where we start with our mother, two capital G's. Then the father is homozygous recessive. So if the father is homozygous recessive, they're both lowercase g's, and we can then split those into the individual gametes. Then all we need to do is fill it out. So start at the top and then put that capital G in the two cells beneath it. Do the same for this one. Start at the top and put the 2 beneath it, and then we start on the left hand side here and we put it in the cells to the right, and whatever's here we put in the 2 cells to the right again. So we've got all the possible genotypes there. What we need to do then is write down what characteristic they would show. Because there's a capital G, then that means they are all going to be green. So what we do is in each of those squares we write green, and the last thing we do is we can write down 100% green, and that's it. That's our total marks. So when you're looking to mark it, if you got this bit, so you got the mother's different gametes, 1 mark. 1 mark for the father's gametes. You can have 1 mark if you got all 4 of the genotypes. So 1 mark for the genotypes, 1 mark for the phenotypes if you got all 4 right, and then 1 mark for your final statement about how many there were. So in total it will give you a total score out of 5. So write your score out of 5. Okay, question 2 for you to have a go at now. Again, show all the working in your books. Okay, so let's have a look at this second question then. Green pea pods are the dominant characteristic, yellow pea pods are recessive. So same as before. Work out the possible offspring for a genetic cross of 2 heterozygous parents. So what we're going to start off with is our Punnett Square once more. You never know, one time I might draw these and they might look a little bit more level. So we're going to have the mother at the top and the father to the side. So if they're heterozygous, then that means they've got 1 dominant and 1 recessive each. So we can then put our gametes in place. So 1 dominant, 1 recessive, 1 dominant and 1 recessive for each parent. And then we just fill out our Punnett square. So whatever's at the top of the mother, we fill out into the 2 squares beneath. Same for this one, we'll fill out in the 2 squares beneath. And whatever is on the left for the father, we then fill out in the 2 squares to the right. And we just put those all in. Then what we need to do is write down what would the phenotype be. So this one's got 2 capital G's, therefore it is going to be green. That one does have a capital G, so it is going to be green. Capital G there, so that one's green. And this has got 2 lowercase g's, therefore that one will be yellow. So that means that we've got a 75% chance of green and a 25% chance of yellow. And obviously you could write that as fractions, decimals, whatever you prefer. So same as before, when we're marking it, we're going to get 1 mark for this one, 1 mark for that column, 1 mark for the genotypes, 1 mark for all 4 phenotypes, and 1 mark for our overall percentages. So it gives us a total of 5 again. So write your answers out of 5. Third question, again, show your working. So third and final question on these then. Again, green pea pods are dominant, yellow pea pods are recessive. This time we've got a heterozygous father and a homozygous recessive mother. So we can start off with our Punnett Square once more. And mother is going at the top, father going on the side. So our father is heterozygous, so that means we've got one capital, one lowercase, and the mother is homozygous recessive, so both lowercase g's. So let's fill out our gametes. So for the mother, it's 2 lowercase g's. For the father, 1 capital and 1 lowercase g. Then we start whatever is at the top of the mother's column, we fill out into the 2 columns beneath. Same for the other side. And then what is at the start of the father's column, we fill out to the 2 cells to the right. So we'll just fill those in. And then we need to write the phenotypes in. So this one's got a capital in there, so that means it's going to be green because obviously green is dominant. This one has 2 lowercase, therefore that will be yellow, as will this one because it's both lowercase. So what we see in this case is a 50% chance of green and a 50% chance of yellow. So just to remind you of where your marks come from, 1 for the mother's gametes, 1 for the father's gametes, 1 for all 4 genotypes, 1 for all 4 phenotypes, and 1 for your final answer there. And that'll give you a score out of 5 again. So give yourself a score out of 5 for this question. So we've had a look to see how we work out the inheritance of certain characteristics, but there's one specific one we need to know about to finish off today, which is how we decide if you're male or female. So see if you can write down what chromosomes decide if you're male or female. Hopefully we knew that for a male you will have 1 X and 1 Y, and for a female it's 2 X's. So they're the chromosomes that determine your gender. Now what I'd like you to do is jot down those top 2 points first of all, make sure you know that and don't just write down Y for male, it's got to be 1 X, 1 Y, and female 2 X's. Then I want you to draw a genetic diagram to show how gender is inherited. So in this one, we're drawing a genetic cross diagram to show how gender is inherited. So we're going to start off with our Punnett Square again. So let's get it set out. And the mother is going at the top and the father on the side. So the mother is obviously female, therefore we'll have XX. The father is male, so we'll have XY. So let's fill out our individual gametes then. So the mother will have an X in each, and the father will either be an X or a Y. So all we're going to do is fill out the potential genotypes for our offspring. So at the top we've got an X, so that goes into the 2 columns beneath. Here we've got an X, goes into the 2 columns beneath. Start of this father's row is an X, so it goes in the 2 columns to the right. And this one is a Y, so we put a Y into the 2 columns to the right. So what we then do is write down the phenotype. This one is 2 X's, so they are female. 2 X's here, so female again. And this is an XY, so male, and an XY here, and male. And that gives us a 50% chance of male, 50% chance of female. So just remind you where your marks come from. 1 for the gametes for the mother, 1 for the gametes for the father, 1 for the genotypes for all 4 offspring, 1 for the phenotypes for all 4 offspring, and 1 for your actual percentages. Total score out of 5. And do remember that when we're talking about this inheritance, it's not a case of if you have 2 children, the first one is female, the next one will be male. It's a 50% chance in each and every pregnancy. Last thing for today then is just a couple more questions on our genetic cross diagrams. So I'll leave these up for a couple of minutes, but as always, if you need longer, hit pause and take as long as you need to answer them.